This is Aditya Kumar Bajpai, and I will be presenting on the topic of analysis of Alzheimer's variant in embryonic stem cell derived microglial cells. So, first of all, the basics about the Alzheimer disease. It's a brain degenerative disorder of unknown etiology, and it is most common form of dementia that usually starts when a man is in its late middle age or an old age, which also results in memory loss and disruptive thinking and change in personality, mood, etc. Now, these degeneration of brain neurons especially happens in the cortex region, the cerebral cortex region, and preference of neurofibrillary tangles. Now, the Alzheimer's disorder is like not very well studied here and have impacted different types of diseases. When we look into the genetic factor which contributes for the Alzheimer's disorder, there are various cell types which we study, like astrocytes and microglial cells are very crucial to study. And we have done a genome wide association study which have identified numerous polymorphism like which elevates Alzheimer disorder conditions and causes a high risk of getting Alzheimer's disorder. Now, the study has been done by a group of scientists, and they have published their dataset with the accession ID as given in the slide. And in that accession number, they have taken several samples of some of the Alzheimer disorder variants which are present on the loci CD33, INPP5D, SORL1 and TREM2. So what are these? They are basically transmembranous proteins or some sort of membrane related components which perform different functions and are active for proper working of the neurons and the brain itself. Now when they get disturbed, we get certain conditions which rapidly accelerates the condition of Alzheimer's disorder. So in this data set we have divided our samples according to the table shown below in the PPT. Now small information about these types of variant. Now these are isogenic human derived microglial cell lines. The variants are introduced in these cell lines, and we see certain type of interesting things like the SORL1 microglial like cell lines induce TREM2 expression which enhance the APOE expression pathway. Now the Alzheimer disorder associated SORL1 and TREM2 mutations also impaired the microglial cell lines alpha beta uptake in the APOE dependent manner in vitro, of course. The CD33 TREM2 and SORL1 are all types 1 membrane components highly expressed in the microglial cell lines. When we have to characterize the effects of Alzheimer disorder associated variants in the microglial cell line, we integrated all these mutation into the H9 ESC line by CRISPR-Cas9 editing. And by we here I don't mean I mean the scientists who have written the research paper. Now the methodology I have adopted to study the dataset they accomplished is basically, first of all, we have to convert the TPM values to raw counts. So preparing the raw counts by batch filtering and other methods and doing a quantile normalization for them. After that we analyze all the raw counts and do a PCA analysis for all the different samples and all the different sets under different condition. We compare all those variants separately and together with the wild type to get a better idea of what's happening with all those different variants. Then we do a gene ontology and KEGG enrichment pathway analysis, which determines the overall direction and the pathways involved in the regulation of those gene samples. Now we derive the volcano plot to find the log to fold charge versus the p-adjusted value plots, and then we finally reach to the conclusion of which type of genes are being upregulated or downregulated, and furthermore results. Now coming on to a PC analysis pipeline, so we have used the tau-info browser provided by our Omics Logic website to do all our work, and as shown in the diagram in the slide, we have adopted this type of pathway to perform our PC analysis, and we provided our raw counts, then we performed our quantile normalization, and then a PCA function was added to it which was being used, and we got our results. So here are some of the charts. I will not gloss over them in detail, but they are comparison of every variant of Alzheimer's disorder with respect to the wild type. So as you can see, here it is. Now the interesting part is that we do not see a very clear demarcation or grouping in any of these slides except the TREM2 one versus the wild type. Now in here I would like to draw your attention towards the TREM2KO type of variants, which is very differently grouped from the TREM2R47H and the wild type, which are grouped together. So that is an interesting finding in the paper. I have written in the results also, and we have worked on that too. Now this is a PTA scatter plot, PCA scatter plot for all the variants together, and you can see that there are two different grouping here. So yes, this is a very significant result, and we have found approximately we have developed this PCA from a pool of 60,000 sample size genes. After doing that and getting the PC analysis done, we have performed the differential gene analysis and the gene ontology enrichment pathway, where we have again used this type of pipeline, and the most important thing is we have kept our p-adjacent value of less than 0.05 and our log to fold change value of greater than or less than -1, which we have achieved here, and we have reduced our significant gene pool from 60,000 to less than 1,300. And the results of DEG analysis are like this, and you can see that most of the pathway which are activated are like the amino acid biosynthesis process. These are related to the biosynthesis and the catalytic processes here. The hormone metabolic process, iron hematostasis, and cellular amino acid metabolism pathway, the carbolic acid or organic acid, carboxylic acid biosynthesis pathway, and the suppressed paths are like signal transduction and response to hormone, biological adhesions, cell dedifferentiation, and cellular developmental process, the tube morphogenesis. This type of pathways are suppressed here, which ultimately signifies that there is a suppression in like extracellular linkage between the cells, and there is not a very proper communication or type of structure which can support neuron and nervous conduction. Now this is a different chart here, and you can see we have taken some of the major significantly affected pathways here, and you can see almost all the major pathways which are associated with the Alzheimer disorder are being suppressed here. Now in the volcano plot we have found out that most of the significant genes here are significantly reduced in terms of their expression. They are between -2 to -4 in terms of log to fold charge, and the p adjacent value is also very less. This is a second plot which we have drawn, and we found out that these 8 genes are very much downregulated and are considered to be affecting our Alzheimer's degenerative disorder and lead to very rapid progression of Alzheimer disorder and onset of it in a very early stage. Now, the conclusion down here was that the TREM2 loci, as we have seen, the pathway which is suppressed and is related to the structural organization of the actin fiber, and the SORL1 loci generally downregulates growth and communication, as most of the pathways suppressed are signal transduction or cell proliferation. The INPP5D loci, it usually downregulates the cell repairing process and the proper functioning of the cellular activities, while it also causes some disruption in the immunity signaling and causes immune distress signal to propagate within our cortex structure. Now, these variation all points towards the degradation in the condition from the wild type genes, which actually are our control group here, and wild type signifies that of a normal individual. So we can clearly see that these type of variations in the Alzheimer's microglial cells can induce Alzheimer's disorder easily and an early onset of it in all the individuals. So thank you for listening, and these were the main findings of our project.